thank you everyone for coming to my talk today on why do we age. Aging is a really mysterious topic that scientists have tried to unravel over the years, and the aim of this talk is more or less, just to give you a brief outline of how the aging process actually takes place, and I'll also be looking at the possibility of immortality, and I'll also be taking a look at progeria, a disease that actually accelerates aging. I'm just going to start off by telling you guys where I got my inspiration to actually do this project. For me personally, it came during my time volunteering at my local care home. And during my time there, I had to interact and communicate with old people, and this involved playing Scrabble, reciting poetry, and doing a bunch of other stuff, which I don't really want to mention. And yeah, basically during my time there, I think I just gained an understanding of how the old people feel about the world, and I just asked myself the question, is it really worth getting old, going to the 90s? You have to face a lot of diseases, you have a lot of social problems, such as depression and others. And I think it just kind of clicked for me right then, well, why do we age? The process is something I never really thought about. It's not a question anyone's ever asked me, or a question I've asked anyone else. And I've also had a look at our AS textbook. It never crops up anywhere, and the answers to it aren't really anywhere from where I found. So I've done this research just to inform you guys of this complex process. Well, as you all know, aging is an inevitable factor of life which mankind over the centuries has actually tried to stop and prevent, but as you all know, it's failed really badly. And aging in human beings brings about multiple physical, psychological and social changes, but at the end of the day, it always leads to death. And the main focus of this presentation is going to be looking at the biological process of aging, and the scientific term given to this is senescence. And senescence is the accumulation of changes to the molecular and cellular structure of an organism over a passage of time, which eventually leads to the deterioration and death of an organism. And basically, I really feel that senescence is an amazing process because as we grow older, not only our features change, but each and every cell in our body is aging, dying, and the cell cycle keeps happening every single moment of our life. And senescence is amazing because it not only happens on the organism as a whole, but it happens on a cellular level as well. And at the moment, scientists are doing intense research on this process, and they're trying to find out whether senescence as a biological process can actually be halted, prevented, or even reversed, which goes into the topic of immortality. And before I actually go on to the next slide, I'd just like to tell you guys that at the moment, scientists actually have no idea why we age. So I'm really sorry if I expected to find out a true answer. But what scientists have done is they've actually proposed several theories to this, which are more or less make up the reason of why we age. So as I said, the scientific community can't actually agree why humans age. So what sort of cellular and molecular biologists have actually done is they've examined research done alongside the aging process, and they've proposed several theories and hypotheses. And they can be split up into two categories, which are the program theories and the error theories. And the program theories basically says that aging has a biological timetable or a time clock, and this basically means that the day we're going to die has been predetermined, and aging has got to do with the genetics of our body. As you all know, DNA is what makes us up. And without DNA, we wouldn't be who we are today, and DNA makes us different from every single person in this room. And as what they've proposed is, in program theories, is that genetics makes it seem like it determines each aspect of our life, such as puberty, the menopause in women, as well as death. And a few examples of the program theory is, and this is probably the most famous theory, the telomere theory. You also have the endocrine theory and the immunology theory. I'll be talking about the telomere theory later on in this talk. And I'll bring that aside. We have the error theories, and they simply say that aging is the process of accumulated damage to our cells and organs over the years. As we grow older, our cells and organs keep on getting damaged and damaged, and this leads to increased immunity, thus making us more vulnerable to diseases and eventually leading to death. And a few examples of the error theories are the DNA damage theory, which I'll also be going into more detail later on, and radical cell theory. Before I go into the telomere theory, there's something we need to understand, and this is called the Hayflick limit. Or I think it was in 1961 in some lab in America. You see the picture of this guy. He's called Leonard Hayflick, and he and this person called Paul Moorhead, they discovered that cells can only divide a certain number of times, And the coined this word, just cellular senescence, it basically means that cells in culture can only divide 50 times on average. And that was really surprising for me because I thought that cells were immortal and they kept dividing constantly until they actually died. And so they called this the Hayflick limit in recognition of this person, Leonard Hayflick. And this term is also called the phenomenon of growth arrest after a series of cell proliferation. And what Leonard Hayflick and Paul Moore had actually did was they created this graph to show the different phases of a cell in a cell cycle and in cell culture, and they split them up in phase 1, phase 2, and phase 3. And phase 3 is cellular senescence. Phase 1 is just a period where cells divide rapidly and cover the cell culture. And phase 2 is when you actually do a process known as sub-cultivation, when you take the cells from one culture and transfer them to another one. And this basically makes the cells divide rapidly. They keep dividing rapidly for like several days, like 210 days here. And towards the end of phase 2, they actually stop dividing. And that's when phase 3 enters. Phase 2 is actually called the period of luxuriant growth by Leonard Hayflick. And phase 3 is cellular senescence, and that's when the cells go into growth arrest. And basically what happens is once the cells stop dividing completely, they undergo something called a programmed cellular death. I'm sure most of you guys are familiar with it. It's called apoptosis, and it is essentially the same as cell suicide. Well, there are a few ways to detect cellular senescence in culture, and they're called biomarkers. And a biomarker is basically the measure of a biological state. And the four main biomarkers are growth arrest, cellular morphology, the enzyme beta-galactosidase, and telomeres, which I'll be going into more detail later on. Growth arrest is probably the most obvious one. If cells stop dividing, well, it's just going to be growth arrest. And what scientists have discovered is that in a cell culture, it's heterogeneous, meaning that there's a small proportion of cells which are senescent, which means they're not dividing anymore, but there are also cells which continue dividing. And basically in a population where all the cells are senescent, it means that it's said quiescence, I believe. And I'm showing you AS levels, you must have learned with a cell cycle. You have G1, G2, and S, so growth 1, growth 2, and synthesis. And growth arrest actually takes place between G1 and synthesis. And the next one is cellular morphology. I'm sure you guys can see these two cells. Basically what happens is as a cell continues dividing and reaches 50 and it stops dividing, the size and the structure of the cell actually changes. So it goes from something as small as that to something as large and big as that. So the structure and the shape of the cells actually change. And what scientists have discovered is that in a cellular senescence culture, there is actually a lot of diverse morphotypes. And the third one is going to do with the enzyme beta-galactosidase. And this enzyme is the lysosome. So it breaks down cellular junk into material which the body can use later on. And this enzyme basically, scientists have noticed increases in cells as you become older. And they also found this positive correlation between the number of enzyme beta-galactosidase and cellular morphology. So the more morphotypes, or the larger the cells, the more weird the cells are, the more they found the enzyme beta-galactosidase. And the final one, and probably the most important one, is telomeres. And telomeres are repetitive regions at the end of chromosomes. So now I'm going to go into more detail about the telomere theory, which is probably the most famous aging theory at the moment. And scientists are doing a lot of research related to it. And as I said, telomere is a region of repetitive nucleotides at the end of a chromosome, which prevent chromosome fusions and the deterioration of the chromosome as well. And in order for me to explain this, I'm just going to go over the topic of DNA replication once again, because what we've learned in AS level textbook is actually completely wrong. So I'm just going to do a short diagram to explain this to you guys. Well, as you all know, at the first part of DNA replication, the DNA strands split apart because the DNA helicase enzyme breaks the hydrogen bonds between the bases. So you have like one strand and two other bases. And what we learned is you just have 3 nucleotides going to bind to them by the DNA polymerase enzyme. But what actually happens is something known as an RNA primer, and you guys know if you learn more about this in A2 level. An RNA is just a short strand of nucleotides, not really a DNA thing. Basically, this RNA primer, it doesn't attach, it attaches to one part of the DNA strand, but it doesn't attach right to the end, so it comes around there. So it leaves the space there. And from there onwards, the enzyme can actually start binding large loop structures called T loops or telomere loops. And these T loops are held together by several proteins, which are collectively known as the sheltering complex. And the two factors, telomere repeat binding factors, which are very important in this, because the loops stabilize the telomere and they prevent them from being recognized as DNA damages. And this is something which is very important in how it is linked to aging. Well, basically, telomeres, without telomeres, our genes will not be, there won't be DNA replication essentially. We lose genes, and without genes we can't code for proteins, and that just means our body won't function properly. So in a sense, they are just disposable buffers, and they're very, very important. And essentially, the Hayford limit is when telomeres shorten at each cell division, and keep shortening each time a cell divides. And once the length of a telomere goes to 0, the cell can't divide anymore, and then they die as a result. So telomeres are essentially a mitotic clock, because once they run out, our cells can't function anymore, and they die as a result, because they're not useful for the body anymore, are they? So the body programs them to die through apoptosis. So you're going to ask, how can telomeres actually lead to aging? And this is another topic that scientists are doing deep research into at the moment. But what scientists have actually found out is telomeres actually keep shortening as cells divide. They start to unravel, and this can be seen as a break in the DNA. And the body, as a result, sees this as a mutant cell, whatever. So they tell the cell to do apoptosis, which is cell suicide. So essentially, as we grow older, the amount of old cells increases, so more and more cells die due to apoptosis. And this is extremely important because in our body, aging is caused due to the depletion of stem cells, essentially. Because without stem cells, our body's other cells can't be replaced, because stem cells, as you all know, are cells which can divide and become any other cell in our body given the conditions. And the term for that is totipotent, which is something you learn in biology sooner or later. And essentially, once our stem cells get depleted, they can't replace other cells in our body, which leads to us aging. And what scientists have also found out is that people, the oldest people in the world, centurions, people over 100, have the longest telomeres. And they've also found that people with heart disease, liver cirrhosis, osteoporosis, a lot of diseases actually have shortened telomeres in comparison to other people who are healthy and normal. In my opinion, I feel that this theory has a lot of basis to it, because they've done a lot of research towards it. And in a sense, it does make well, but most of the theories we're going to look at actually are a combination of the others. And now we're going to have a look at the endocrine theory just briefly. And this theory basically says that the biological clock in our body is affected and controlled by the hormones. And there is a lot of facts to back this up, because hormones do control our growth, metabolism, temperature, inflammation, stress, and a whole load of other factors which affect our body. And it is the hormones insulin, which is very important for the maintenance of glucose, and hormones such as estrogen and testosterone, which result in puberty in males and females. And also for menopause in women, it is the depletion of estrogen and progesterone. And basically what this theory says is that as we grow older, the control of our hormones decreases, and hormones are very important in the process known as homeostasis, which is the internal maintenance of our environment. And if there was no homeostasis, we won't be living at the moment. And hormones are very important components of it. And without hormones, we'll become more and more vulnerable, because homeostasis won't take place really well. And the final program theory I'm looking at is the immunological theory. And this is quite simply says that our DNA programs a decline in our immune system as we grow older. And essentially the two main cells which are important in our immune system is the T and B lymphocytes, so the T and B white cells. And they're very important because they hit our cells which are invaded by viruses, and they also destroy toxins and antigens and all those other bunch of harmful stuff to our body. And basically what the immunological theory says is that we just become, our immunity just decreases as we grow older. Well, now I'm just going to talk through the DNA damage theory, and this is an example of the error theories. And this theory more or less proposes that aging is a consequence of unrepentant accumulation of naturally occurring DNA damage. So when our cells replicate, or just when the DNA is just present, our DNA is exposed to damages from harmful chemicals, from loads of sources, And our body has a way to have these DNA repair systems which actually fix these breakages, but sometimes these breakages aren't actually noticed, and this DNA damage actually keeps on accumulating over the years, and this results in aging apparently for some reason. And I think it's important to understand the two different types of DNA damage. One is the direct DNA damage, and that just means a physical alteration in the DNA strand. And a few examples can be a loss of a base, just a break in the DNA strand, or a change in the chemical structure of the bases. And that can be subdivided into endogenous and exogenous damage. Endogenous is when it's from an intrinsic source, so just a chemical produced by the cell itself, which could be harmful to it. And exogenous is when just from UV radiation, toxins, and from viruses and mutagens essentially. And the final damage is by DNA mutation, a term which I'm sure you're all familiar with, and that's just a restructuring of the arrangement of the DNA bases in the DNA, and this is caused normally during replication errors. The main thing we need to understand is direct DNA damage can actually be recognized by enzymes, so DNA repair systems can actually heal them, whereas in DNA mutations, if it's on both the chromosomes, because as you know, our body has homologous chromosomes, and basically if it's on both of them, then it can't be recognized as repaired. So why does it actually lead to aging? Well, what scientists have found out is that as our DNA gets more and more damaged, it leads to more cells dysfunctioning and cell loss, because more cells are programmed to die, and this leads to stem cell depletion. And as stem cells deplete, it's very much similar to the telomere theory. So without stem cells, our normal body cells which are destroyed due to infection and other stuff can't be replaced, and as a result, it also leads to a disruption of tissue. And at the end, with less cells, homeostasis is also messed up because we can't produce the necessary proteins to do it. And I think it's the evidence for this theory, which is what I really liked. And what scientists have really cleverly said is that if the DNA damage theory is the cause of aging, then people who do have a defective aging repair system would age at a much faster rate. So what scientists have done is they've had a look at people who have progeroid syndromes. And for those who don't know what those syndromes are, it's when people age at a much faster rate. And a few examples of this diseases are progeria and Vellore syndrome. And basically what scientists have said is, well, if these people do have a defective system, then it more or less means that DNA damage is the main cause of aging. So they've taken down a few trials on people with Vellore syndrome and progeria, and they've actually found a link. They found that people with Vellore syndrome do have a defective system, thus making this theory really plausible. And I think to provide further evidence for this theory, scientists have also done some tests on mice, and they've actually injected bad repair systems into them. They've manipulated the DNA so that the repair systems aren't really effective, and they've noticed that these mice do age at a much faster rate in comparison to normal mice. And I think now that we've had a look at the two most famous theories of aging, I'm just going to have a look at the possibility of immortality. Can I just have a show of hands for anyone who thinks they would like to be immortal in the future? Okay, so there's not a lot actually. Well, as you all know, immortality is just the ability to live forever or to have eternal life. But a major misconception is that if you're immortal, you can't die of any other means. It just means that you won't die due to natural death. It means that if someone knifed you or threw a grenade at you, you still die. So what you were thinking was probably invincibility, and immortality is a totally different concept. And I think over this century, millions of years, mankind has always tried to find a possibility for immortality, but it still hasn't succeeded. But I'm going to be focusing on the scientific side of immortality, and scientists have used the term immortality for cells which they don't do the Hayflick limit, meaning they can divide infinite number of times. And actually, before the Hayflick limit was actually discovered, there was this surgeon, he was called Lewis Carroll or something, and he said he had evidence to prove that cells are immortal, but no one could replicate his results, so that theory is disapproved. And there are a few cells in our body which are actually immortal, and they can be split up into two main types. There's germ known as telomerase, and this enzyme basically ensures that the length of the telomere keep increasing so that they don't shorten at all. So these cells can also divide infinitely. So I'm also going to be having a look at one possible way mankind has tried to gain immortality, and that's cryonics, from the word Greek word cryos for icy cold. And cryonics is basically the preservation of humans who cannot be sustained by contemporary medicine in an ice cold capsule, with the hope that in the future they might be revived and healed using modern medicine, essentially. And basically the basis of cryonics is the fact that the three things can be maintained in people in a cryonics chamber. That is identity, personality, and long-term memory. Scientists have somehow proved that these can be stored in cells in the brain, and basically if you do revive a person in the future, then possibly they might come back to the person. But at the moment, I'm sure you've seen in movies, like if someone's fingers get chopped off or something, you can actually put it in like in ice and actually take it to the hospital and reattach it. So that's probably the smallest form of cryonics which is actually possible at the moment. And the father of cryonics is known as a man called Robert, and he actually died recently. And he created something known as the Cryonics Institute in around 1976, where people who pay around $28,000, you get put in this capsule with liquid nitrogen at a temperature of around minus 320 degrees Fahrenheit. And he himself is now in a capsule with his mother and his two wives for some reason. I don't know why. Well, yes, one of them. He is known as the father of cryonics. And the next slide, I'm going to be talking about immortal animals. At the moment, scientists have only discovered two organisms on Earth which are immortal. One of them is the Venerian flatworm, but I'm going to be focusing on something known as the Hydra. The Hydra is a super organism in the sense that it can't die, and if you hurt it, it'll regenerate its tissue. And the Hydra, if you look at it, it's just a really small freshwater animal. It's got like a radial symmetry with like tentacles coming out of its body, and it's found in lakes and streams around the world, mainly in tropical and temperate conditions. And biologists are extremely interested in this organism because, as I said, it doesn't age, and it can't really die. And that's because it has this amazing super ability known as morphalysis. And morphalysis basically means that it can regenerate its tissue from pre-existing tissue. So, for example, if I get a Hydra and splice it in half using a knife, well, the two halves will actually grow to become two independent Hydras. And this is just something really amazing, because it's just using its pre-existing tissue to come back to life. And this is something I actually didn't believe, and scientists have proved that if you take a Hydra and actually chop it into a million pieces, and you put it in a certain solution, you can technically produce a million Hydra, and this superhuman ability is what scientists are really after. They feel that they can crack this code, maybe immortality is possible. I'm just going to finish off Joey Harry Potter fans out there. I'm sure you know what this is. The Philosopher's Stone. Well, the Philosopher's Stone is a legendary alchemical stone which people thought could convert base metals such as lead into gold. But it also has the amazing feature that it's capable of producing the elixir of life. And some people thought that if you drank the elixir of life in a special cup at a special time of the day, you'll get immortality. And this is probably going to be the last slide, and I'm just going to be having a look at progeria. Well, I was first introduced to this disease when I actually watched the movie. There aren't too many movies on this, but yeah. But well, progeria is an extremely rare disease. Actually affects 1 in 8 people, and people with the disease age at 8 times the normal rate. So the average lifespan of a person with progeria is around 13. And then children with progeria, as they age, they gain diseases such as arteriosclerosis, osteoporosis, stroke, and most of them eventually die at the age of 13 due to a heart attack. It is a really sad disease, and they are doing a lot of research to actually find out what causes progeria. And what scientists have actually found out is that progeria is caused due to a mutant gene. Normally there's a gene called lamin A, and this produces the lamin A protein, and this protein is very important in the structural scaffolding of the nucleus. So that's a normal nucleus, and that's a nucleus of a person with progeria. So basically this protein, lamin A, makes sure that the nucleus is really strong and it's functional. And essentially, if there is a mutation of this lamin A gene, it leads to a different protein being produced, and that protein is called progerin. And this protein can't really do the same job as lamin A, so the nucleus isn't really stable without the structural scaffolding. And essentially, well, this somehow elucidating, and scientists are still trying to continue finding the actual reasons for it. And I'd just like to thank you guys for listening to this, and I'd like to thank Clio and Hutchins Society for providing me this opportunity to give this talk. Any questions?
Right, I actually can't remember the name of this type of jellyfish, but I've heard there's a jellyfish that can actually make themselves back into almost like a child age, like kind of like almost like non-age. Like, have they, like, what kind of research have they done into this kind of jellyfish that can turn themselves back into a child?
I'm not really sure about that, so I can't answer that, unfortunately.
Sorry.
On the subject of immortality, is it possible to like chop off my head and then like put it in like a solution that contains like all the necessary nutrients needed for life? So like a sort of golden solution you make.
That's more of a science fiction thing. Is
it possible to do that? Like, would it work?
I mean, to be fair, you do need the brain to survive, and the main thing of the heart is to provide oxygen to the brain. So it might be plausible, but I mean, really difficult. I mean, what are you going to do just being in a job? What's the point? Yeah, that's...
with progeria, you showed the difference of the nucleuses, the change. Does this particular example lend itself to any of the previous answers to why we age?
I think scientists are really interested in this because they feel that if you find out why we age faster, then you'll probably find out the actual correct answer to why we age. And I think this is a bit different because you have other accelerated aging diseases such as Vellore syndrome, like I said. But progeria is quite unique that it's caused by this factor of the protein, whereas those, like Vellore syndrome, is caused by a defective repair system. But the difference is people with Vellore syndrome live up to 50, whereas this is only about 13. So scientists are still doing a lot of research, and only recently there was a progeria-like organization actually created by people affected by progeria to actually do research into this. So it's only on its early days, so maybe in the future, near future, they might find something more useful.
What about the Benjamin Button thing with Versace?
Sorry?
Benjamin Button thing.
Benjamin Button?
And he's like, he like born old and then died young.
I know that.
On cryogenics, given that almost every cell in the body contains water, and water freezes and expands, does that mean that if you freeze somebody's body, the entire cell structure of the entire human body will change? So wouldn't it make it almost impossible to provide
that is a misconception people have, but you don't really use water as liquid nitrogen.
I mean, but water in the body will expand.
Water in the body, for some reason it doesn't. I think it's just a solution that somehow prevents it from actually happening.
Is there any way to increase the length of the telomere indefinitely to something close to immortality?
No. The only way to have immortality is to constantly keep increasing the length of the telomere, because there's going to be a limit, isn't there? The limit when you're probably born. And like I said, the enzyme telomerase, which some cells in our body can produce, but not all of them. So if technically we can achieve immortality, if every cell in our body can make that cell, that the specific enzyme.
Okay. Yeah, so another big problem with mortality, obviously, with the whole cell issue is about that there's a hard limit to how much our brain can do, how much memory can store.
Yeah.
So the neurodegenerative disease such as dementia. So do you think that probably the thing being a problem to mortality than simply cell death and this process?
Sorry, can you
Do you think the problem of our brain having like a hard limit in how much memory it can possess is a bigger problem for mortality than the problems you raised?
Well, that's possible, but if you think about it, as you grow older, you forget stuff like in the long term, in a sense, so maybe it kind of makes up for it. Because I don't remember what I did last year at this moment. I can't remember what I did like 2 days ago. So I mean,
to manually transfix telomerase as you start to age?
I think the only way you can't do it on a proper human. I think if you create a baby, that comes to the design a baby probably ethics. Maybe if you actually make a baby in a test tube or something, possibly you can manipulate his DNA so that the original, like the sperm and the egg meet, that all the cells from the original embryo can actually make telomerase from the beginning. That's probably the best way. I don't think you can just insert a gene to make the thing, but that's probably the best way. Any other questions?
You talked about like the animal in water that
Yeah.
grows up. So does it have stem cells at its surface that regrow, or do the cells become stem cells?
It's like a super animal really, because it has infinite stem cells or something. So the stem cells just keep regenerating like in the body. But the fact is, it just uses pre-existing tissue. I think nearly all of this tissue contains stem cells for some reason. So even if you cut it into a million pieces, you just make a million of them instead of just like one.
What happens to a grenade at that?
It's immortal, it won't die. Because I mean, unless it gets vaporized, it's probably come back to life, I'd imagine.
When you say that a hydra can't die, wouldn't it overpopulate the ocean? Like Disney cannot die.
Surely there's like a
They're like microscopic.
There's got to be a way that you
No, I mean, they can die of disease. I mean, like that. They can't. But I don't know. That's just something that I'm not really sure of, because from what I've like researched, they said that they can't. They don't seem to die of old age, so they can die of disease essentially.
I think I know the answer, but is there no way without drastically altering the human biology to replicate the Hydra's ability?
There is possibly a way in the future, but I mean, like you know how you take a gene from a fluorescent jellyfish and put it in a rabbit, you can't do it on a human being at the moment, can you? It's more or less the same thing. Once you crack the techniques for doing the simplest stuff, you can move into these more complex DNA techniques, and I think it will be a possibility like in maybe the next 100 years, but probably not at the moment, because you also need to understand how the Hydra actually is able to do this. And what scientists have discovered is that they have this special transcriptional factor called forkhead box or something. And basically these factors are they're proteins which are very important in the gene expression for longevity, proliferation, and differentiation. So they need to improve understanding of how the Hydra actually functions before we can actually use it in humans or use their genes in human beings. Any other questions?